Hi Year 11s and 12s. Today we're looking at the second PowerPoint in the DNA and proteins topic of Stage 2 Biology. This PowerPoint will be broken down into two videos. The first looking at what a gene is and how transcription and translation work in order to create proteins, so protein synthesis. And then the second video will be looking at the structure and function of proteins. We'll start as per usual with our need to know. So these are straight from the Stage 2 outline. These are the things we will cover in this PowerPoint. Again, as per usual, I'm not going to stop and read these out. If you'd like to write these down, which I'd strongly encourage, I would encourage you to pause the video and write these down now. These cover both videos, so there won't be another one in the next video. These cover everything in this PowerPoint, so across the next two videos. So first off, what exactly is a gene and how does it contribute to protein synthesis? Well, a gene is a unique sequence of nucleotides that will code for a protein or an RNA molecule. Okay. One gene can contain hundreds to millions of bases. Each gene will be a different amount of bases depending on what protein it's going to create. Whereas a chromosome contains hundreds to millions of genes. So each chromosome is made up of millions of genes, and each gene is made up of millions of bases. Genes in eukaryotic cells contain segments that code for proteins, but they also have sections that code for nothing. They're non-coding sections of that gene. We have names for each of those. Exons are the segments of a gene that code for a protein, whereas introns are segments of a gene that don't code for a protein. And we'll talk about how we separate these two from each other in this video. So, what is a protein? We know how a gene creates a protein, but what is a protein? A protein is a large molecule that's composed of smaller subunits called amino acids. Okay, so again, like we talked about last week, a protein is a polymer, and the amino acids are the monomers, and they're being built up together to build that protein. Okay. The structure and function of the protein is all dependent on the sequence of the amino acids in that protein. So, with only 20 amino acids found naturally in the environment, they can appear in a number of different orders, and whatever order they're in and the sequence they're in will determine the function of that protein, because the sequence of those amino acids determine the shape of the protein, and the shape determines the function. The sequence of amino acids in the protein are all determined by the nucleotide bases in the gene. So whatever bases are there, that will determine what the amino acid sequence will be, hence it will determine the structure of the protein, which in turn describes the function of the protein. So when we're actually creating a protein, there's two different stages that we need to know: transcription and translation. And again, like I said before, biology is all about the words that we use. So transcription, transcribing something, you're copying something. Translation, you're translating something, you're turning it into a different language. So in transcription, you're copying the information that's in the DNA or in the gene onto a messenger RNA molecule. Okay? As we discussed last week in class, that's because the DNA is too important. We can't have the DNA leave the nucleus. It needs to be kept safe at all times. Whereas the RNA, we can safely make a copy, send it outside the nucleus to create a protein, and we don't need to worry if that's damaged because the DNA is still secure and the original copy is still secure in the nucleus. Okay. Now, when we create that mRNA in transcription, we're copying everything, the introns and the exons. Now, we only want the exons. The introns don't code for proteins, so we need to get rid of those, and that happens through RNA splicing, which we'll look at in this video in a later slide. Once that's happened though, so once we've got a copy of the DNA and it's now a messenger RNA, and we've removed the introns, we go to translation. Because proteins are made up of amino acids, but we have a messenger RNA here that's made up of DNA nucleotides. Nucleotides. So translation means we're translating it from the nucleotides into amino acids to create a protein. So, we're going to look at each of these now in order. So transcription first, and then we'll look at translation. We'll also look at RNA splicing in between the two. So transcription occurs in three different stages that you must know. So stage 1, the weak hydrogen bonds are broken between the DNA strands by the enzyme we talked about last week, helicase. That causes the two specific polynucleotide strands to separate at one specific point of the DNA molecule. So when we talked about DNA replication last week, we said helicase was unzipping the entire DNA strand and separating the two strands entirely. In transcription, we only want one specific gene, so helicase will only unzip the DNA molecule at the site of that specific gene. The rest will stay zipped up. Once that specific section is unzipped, we move to stage 2, where the free RNA nucleotides in the nucleus come in, they bond to the complementary bases on the strands of the DNA molecule, and they form those weak hydrogen bonds with the exposed DNA strand. RNA polymerase will then join the sugar and phosphate groups of those adjacent RNA nucleotides, creating a sugar-phosphate backbone. The mRNA strand will then detach from the template strand, and it can then leave the nucleus and make its way to the ribosome in the cytoplasm. We've got a video of that happening. Okay, so we've got a couple of different videos here. We'll just wait for them to restart before we actually talk about them. As you can see though, this one's the best one, not the entire DNA is unzipped. Only a specific section where the gene is. Once it's unzipped, we're placing those free nucleotides down next to the complementary base pairs in the DNA, and we've got this connecting up the sugar phosphate backbone. So once we've done transcription and we've now got our mRNA strand, like I said before, we need to remove the introns so that we've only got the exons left. The exons are coding for the protein, and they're going to tell us what amino acids to put into the protein, whereas the introns are basically, like I said before, non-coding segments. They're not needed. It will just confuse the whole situation. So we need to go through RNA splicing. This is the process that removes those introns from the pre-mRNA. So when it's straight from transcription, we call it pre-mRNA. And once we've done RNA splicing, we call it mature mRNA. That mature mRNA will only contain the exons. Splicing is carried out by a protein called spliceosome. Spliceosome goes along that mRNA strand, and it will physically cut out the introns, leaving only the exons, therefore allowing us to create the protein. How does this all work though? How can a single section, a single gene, tell us exactly what amino acids we need and what protein we're going to create? It's all done through the genetic code. The genetic code helps to translate the nucleotide base sequence from mRNA into amino acids in a protein. Every 3 nucleotides in that mRNA strand are called codons, and each codon codes for 1 amino acid, or a stop signal telling the protein that it's finished. There are 64 codons, 4 to the power 3, and that's because there are 3 bases in a codon and 4 different bases to choose from. If we had only 2 codons, we wouldn't have enough options. We wouldn't be able to cover all 20 amino acids. If we had 4 bases in the codon, then we'd have too many and it would become too cluttered. So we have to have 3 bases in each codon to allow us to cover each of the 20 amino acids, which gives us 64 codons in total, meaning that some codons will go to the same amino acid. Protein molecules can only contain one of the 20 known amino acids. We classify the genetic code as being degenerate because the amino acids can be obtained with those multiple codons. So for example, AGU might code for the same amino acid as UAC. They probably don't actually code for the same amino acid, but just as an example. We also describe the genetic code as non-overlapping, because if one of those 3 nucleotides has been used in one codon, it's not going to be used in the next codon. It can only be used in one codon in each mRNA strand. Here's a copy of the genetic code. So for example, if we've got UCU, UCC, UCA and UCG, all of these code for the amino acid serine. Okay, same over here, we've got GUU, GUC, GUA and GUG, all coding for valine. Okay. just quickly, if you ever have to use that codon, you will have to use that in tests and exams. That will be given to you in the exam. Same with this. This just tells you, sometimes they'll only give you the 3-letter code for the amino acid. This just converts that into the full name. But again, if you need that, it'll be given to you in an exam. You don't need to memorize that. It's just a copy to let you see what it looks like. Now we're moving on to translation. So we're actually turning the mRNA into a protein, actually turning those codons into amino acids. And that's all done at the ribosome. Now the ribosome itself is made up of RNA. It's made up of ribosomal RNA, which we call rRNA. So after RNA splicing, the mature mRNA travels through the pores of the nuclear membrane into the cytoplasm of the cell. It's no longer in the nucleus. That mRNA is now in the cytoplasm of the cell. It will then join with the ribosomes in the cytoplasm, generally on the surface of the rough endoplasmic reticulum. Not all ribosomes are there, but most are, so that's why we say that. Ribosomes, as you know from year 10, are the site of protein synthesis. There's close to 100 million of them in each of your cells, so there's a lot of them to do protein synthesis. Each ribosome is made up of a small subunit, shown here in pink, and a large subunit in purple. The small subunit is responsible for reading the mRNA, while the large subunit is physically joining the amino acids. Ribosomes themselves are made up of 60% rRNA and 40% protein. rRNA in the ribosome will work as an enzyme. It helps the large subunit catalyze the formation of that protein molecule, help connecting the amino acids. Another part in translation is transfer RNA. This is really important. You don't have to draw this structure. You won't ever have to recall this structure. You just need to know roughly what transfer RNA does. So it's a small RNA molecule made up of around 80 nucleotides. So you can see this isn't drawn to scale. And its job is to transfer the amino acids to the ribosome during translation. The tRNA molecules have an attachment site here for a specific, over here, for a specific amino acid. And they also have a section over here called the anticodon, which will bond with the codon. So each amino acid will be attached to a transfer RNA that has an anticodon that's complementary to the codon that codes for that amino acid. Like the codon itself, the anticodon is 3 nucleotides so that it can match perfectly with the codon. Translation itself. Translation occurs in 4 different stages. First off, a mature mRNA molecule, so we've had the introns removed, will attach to the small ribosomal subunit, and the first mRNA codon will be covered by the ribosome. That first codon is going to be read, and a transfer RNA molecule will bring the first amino acid to the ribosome. Hydrogen bonds will be formed between the anticodon and the codon, knowing that we have the correct connection there. The mRNA molecule will then move along so that the second codon is covered, and the second transfer RNA will bring in a different amino acid to that ribosome. And the large ribosomal subunit will create a chemical reaction that will join those 2 amino acids together. Stage 3 is going to be repeated until it reaches the stop codon. That stop codon tells the protein molecule that it's finished. The entire amino acid chain will be cut loose, and the protein will be ready and moved into the rough endoplasmic reticulum so it can be modified and used in the body. We have a video here of that working. So as you can see, we move the mRNA along, transfer RNA brings in a amino acid, the large subunit connects them up, and we keep moving down and adding more amino acids as we go.